Hello guys, welcome back to my channel. Today we are going to learn hydrolysis of esters in organic reaction mechanism. We'll find out hydrolysis of esters. It is very common topic and you might be doing from your graduation. Now you can see here there are two possibilities of hydrolysis of esters. You have seen that it might occur in the presence of acid or it may be in the presence of base. Because of the poor leaving group, it cannot be hydrolyzed in the absence of any catalyst. So we require catalyst here, and the products are acid and alcohol. In the presence of acid as well as in the presence of base, it occurs in the form of salt. Okay, so we'll see some more important aspect here, that so far we used to know that only the mechanism occurs in the presence of either acid or base catalyzed hydrolysis of ester, but actually there are total 8 possible mechanisms, as you can see here mentioned. A for the acid catalyzed and B for base catalyzed. So if the acid is given to you, you have to find out there are 4 possible combinations for the hydrolysis, where AC for the acyl cleavage and AL for the alkyl cleavage. So again, there are 2 possibilities. In the presence of acid, the hydrolysis either the cleavage is acyl cleavage or alkyl cleavage, and in that also you have 1 and 2 means unimolecular or bimolecular reaction, means the rate of reaction, whether it affects by only on the substrate, it depends on the substrate concentration, or it is depending on both substrate as well as the reagent concentration. Similarly, it is in the presence of base, either acyl cleavage or alkyl cleavage. It may be unimolecular or bimolecular. So there are total 8 possible combinations, and the common mechanisms are here SN1, SN2 or tetrahedral mechanism. As it forms a tetrahedral intermediate, SN1 you can easily identify based on the carbocation formation, SN2 again inverted configuration. And we'll see all these mechanisms with the help of problem. This is our very first problem. As you can see, ester is given here in the presence of concentrated acid. 2 question mark means there are 2 products, one is acid, other is alcohol, and this is the hydrolysis of ester in the presence of acid. So this is our question here. Now in the presence of acid, we know there are 4 possible combinations, in that it may be acyl cleavage or alkyl cleavage. Here alkyl cleavage is not possible through SN1 because it doesn't form the stable carbocation. So SN1 mechanism is not possible, and you can see here it is sterically crowded. So we will see the tetrahedral mechanism is also not possible, right? Or we'll find out then what is the next possibility, the acyl cleavage, right? So acyl cleavage, because if it is alkyl cleavage is not possible, then acyl cleavage, and therefore the protonation will occur on this oxygen which is attached to the amine, and it releases as alcohol, leaving behind the stable carbocation. This step is slow and therefore it is the rate determining step, and only the substrate concentration is involved here, therefore it is the unimolecular acyl cleavage. And since it is a stabilized carbocation which is also stable by the 3-methyl group, the nucleophile will attack on the carbocation and it forms the acid here. So what is the mechanism here? It is acid catalyzed acyl cleavage unimolecular, right? Acid catalyzed acyl cleavage unimolecular. Since the carbocation is formed here, the mechanism is SN1 mechanism. So it is easy to identify. Note is the hydrolysis of sterically hindered acid, you can assume it occurs through AAC1 mechanism if the solvent is having the strong ionizing power, like concentrated acids. If it is given to you, you can think of the mechanism AAC1, that is SN1 mechanism. Now if you have this type of ester in the presence of dilute acid, you have to identify, obviously it cannot be AAC1, right? So what is the possibility? Now again, alkyl cleavage is also not possible here, right? Because it doesn't form the stable carbocation. What is left then? Acyl cleavage, and that acyl cleavage is happening through the formation of tetrahedral mechanism, because both sides you don't have sterically grounding also, so tetrahedral intermediate can easily be formed, and therefore in the presence of acid the nucleophilicity of this carbon increases, and so the nucleophile will attack on this carbon, and you can see the loss of this alcohol will occur. The proton will be abstracted by the lone pair of this oxygen, and so it releases as alcohol, leaving behind the carbocation. With the loss of proton, you get the acid here. Since the concentration of this substrate and the reagent is involved in the rate determining step, which is slow here, so it is a bimolecular reaction. So we can easily identify now it is acid catalyzed acyl cleavage bimolecular reaction, which is called the tetrahedral mechanism. This is a tetrahedral intermediate. So this is how you can approach the mechanism. Correctly you can mention it. Now this type of ester, if you have in the presence of acid, you can see this is optically active ester. You have this carbon is chiral having all 4 different groups in the presence of acid. Now if you think of acyl and alkyl cleavage, this alkyl cleavage is more feasible here, because once it releases, it forms a stable carbocation, which is stabilized by all the 3 electron releasing group. So definitely it is the alkyl cleavage in the presence of base. And how you can release this carbocation? With the protonation of carbonyl oxygen, and this alkyl cleavage will occur, leaving behind. So this mechanism is slow here, and only the concentration of this substrate is involved, so unimolecular. Since it forms a planar carbocation, it is a SN1 mechanism, which is a stable carbocation. Now this nucleophile, either it can attack from above the plane or below the plane. So therefore you will get the 2 possible product here, which are the racemic mixture and optically inactive. So you have started with optically active ester, but you are leading with the optically inactive product here, because you are getting the racemic mixture here. Being the planar carbocation, the nucleophile can attack either from above or below the plane. So this is the mechanism is base catalyzed alkyl cleavage unimolecular. Not base, it is acid catalyzed alkyl cleavage unimolecular SN1 mechanism, right? So this is carbocation. Like that you can easily identify. I hope you can do this problem yourself. This is again a CCH3 whole thrice. After cleavage of this bond, it is going to form a stable carbocation, which is tertiary carbocation. So in the presence of acid, alkyl cleavage will occur the same way like we have done the problem number 3. You can see the stable carbocation, the nucleophile will attack here and give you the alcohol. Since the rate determining step is involved only in the concentration of substrate, it is the unimolecular. So the mechanism is acid catalyzed alkyl cleavage unimolecular SN1 mechanism. Carbocation formation. Formation of carbocation proves that the mechanism goes through with SN1 mechanism. In this, again, you have now, instead of acid, a base catalyzed hydrolysis of ester. You have ester here. In the presence of strong base, now either acyl cleavage or alkyl cleavage, that we have to figure out. If you talk about the alkyl cleavage is not feasible through SN1 mechanism, because that doesn't form the stable carbocation here. And being both sides, you don't have bulky group, the tetrahedral mechanism is easily possible. So the base will act as a nucleophile, will attack on the carbonyl carbon and form the salt and alcohol. Formation, and you can see here the retention of configuration. Here you have S ester, here also you are getting the S alcohol. So retention of configuration you will get only if the mechanism is through tetrahedral. In case of carbocation formation, rearranged product will be there, and in case of SN2 you will get the inverted, right, inverted configuration. So this is a proof of tetrahedral mechanism. So this is base catalyzed acyl cleavage, right? This is acyl cleavage, bimolecular because the concentration of base as well as substrate affect the rate of reaction, tetrahedral mechanism. And this is again, you can make a note, the esters which are having the chiral group give the chiral alcohols with the retention of configuration, right, via tetrahedral mechanism. Now, this is again ester, but here the conditions are mild. You have mild alkaline system here. So here again, if you notice, you have a chiral compound optically active, and if you think of fragmenting this CO bond, which makes carbocation formation, which is a stable carbocation, which is stabilized by pH aromatic ring. So this is base catalyzed alkyl cleavage. You can see here forms a stable carbocation, again either above the plane or below the plane. So you will get the racemic mixture which is optically inactive. Since you are getting racemic mixture, which suggests the mechanism has gone through with the SN1 mechanism through the carbocation formation. So basically it is base catalyzed acyl cleavage, not base catalyzed alkyl cleavage, unimolecular, because this is the rate determining step, only the concentration of this substrate is involved. So unimolecular SN1 mechanism. Like that, you can figure out. Here, notice alkyl group of alcohol is capable of forming a stable carbocation. Therefore, the mechanism goes through SN1 mechanism, alkyl cleavage. And moreover, the solvent is having the high ionizing power, which is the medium is weakly alkaline. Therefore, the mechanism is BaL1. So if you have high ionizing solvent, then you can think of BaL1 SN1 mechanism. Now this again, you can think and solve the problem yourself. You can see here, this is ester hydrolysis under mild condition. Just now I told you, if under mild condition, high ionizing power, you can think of alkyl cleavage, right? This alkyl cleavage bond, if you think, it is going to form a carbocation here, which is allylic carbocation. You can see 3 carbons are present with a double bond, and which is stabilized by this aromatic ring as well. So it is allylic carbocation, stable carbocation. So therefore, you can assume the base catalyzed alkyl cleavage. Base catalyzed alkyl cleavage forms the carbocation, which is stabilized by resonance. So you are getting here 2 intermediates here, carbocation intermediates, with the nucleophilic attack on these carbocation. It forms, one is the normal retained configuration and other is a rearranged product. The formation of rearranged product itself suggests the formation of carbocation. Okay, so if you are getting the rearranged product, that is only possible if the mechanism has gone through with the carbocation formation. So we can find out the mechanism is base catalyzed alkyl cleavage, right? Base catalyzed alkyl cleavage, unimolecular, because the concentration of this only affects the rate of reaction, SN1 mechanism. So I hope now you are getting how to figure out the type of mechanism. And this is going to be the last problem in today's discussion. You can see here you have the isotopic water to identify whether the isotopic oxygen remains with the acid or with the alcohol. Two products, one is acid, other is alcohol. Now you have to look carefully. You have sterically crowded ester here. Now what is the possibility if you have a sterically crowded ester? First of all, you have to find out here that here tetrahedral mechanism is not possible because it is sterically crowded. So the nucleophile cannot approach directly here. SN1 also here doesn't form the stable carbocation either side. So basically it is the SN2 mechanism you can think of, backside attack. So this base will attack through backside and it is the alkyl cleavage which gives you the isotopic water with the oxygen with the alcohol. SN2 mechanism. So basically the concentration of both are affecting the rate of reaction. So it is base catalyzed alkyl cleavage bimolecular SN2 mechanism. Right? So like that you can solve this problem. Now the mechanism is basically which condition you can think of this when you have a weakly alkaline system and the carbonyl group of ester is sterically crowded. If both the conditions are there, then you can think of Bal2SN2 mechanism. So if you make these points as a note, this will help you as a tricks. Okay, instead of thinking, you can directly apply these tricks. So like tricks are in the form of note I have written here. You have weakly alkaline or neutral system and sterically crowded carbonyl. Then you can directly think of Bal2SN2 mechanism and approach this mechanism and solve it. So like that, I hope now after solving so many problems, you have an idea now how to approach the correct mechanism for the hydrolysis of esters. You can solve more problems based on the topic we have discussed today and have a hold on this topic. All the best, happy learning. See you in the next video.